Hi, everyone. Welcome to the second episode of Our New Viral World. I'm Makeda Robinson. I'm an infectious diseases specialist. And today I wanted to talk about the race to a COVID-19 vaccine. There are several vaccines in development, but I wanted to talk specifically about 3 vaccines that are farthest along in the pipeline and are using really novel and interesting technologies. In a typical situation, the development of a vaccine from start to finish can take up to a decade, if not more. And these vaccines go through 3 clinical trial phases. The first phase, or phase 1, is usually performed on a smaller number of healthy people and is focused on the safety of the vaccine. So when you get this vaccine, are there any side effects? And how do the people feel after getting the vaccine? The second phase recruits more people and is also now evaluating if the vaccine works. So after you get this vaccine, are you immune to the pathogen? And in this specific situation, the virus, after you get this vaccine. And phase 3 requires the recruitment of many more people, so definitely over 100, but oftentimes it's at multiple different places so that we know that different people coming from different places will all have the same response to the vaccine and can often be compared to whatever the standard of care is. In a traditional timeline for vaccine development, there's a longer period of time spent by an institute identifying a target for the vaccine and making a go or no-go decision about the candidate vaccine. And once this is done and preclinical trials have been performed, the first trials in human will be performed, and they will go through the 3 phases that I just discussed. So phase 1, phase 2, and phase 3. An important point to notice here is that typically an institute would not begin to sort of stockpile this vaccine or start to manufacture a lot of the vaccine until we're really into phase 3 and closer to the vaccine being licensed, because there is a large financial investment being made in order to make a lot of this vaccine. However, many of these steps are consolidated in the setting of an outbreak or a pandemic, so the situation we're in now. So as you can see here, the amount of time spent upfront on target identification and the decision about whether or not to move forward with a candidate vaccine is much shorter. And in fact, all of the vaccines that I'm going to talk about today have the same target. And additionally, the human phases of the clinical trials often are overlapping. And so we're looking at the vaccine safety and dosing and efficacy in combination. And the decision of an institute to begin to manufacture and develop the vaccine often happens much earlier on so that it is available as soon as possible to as many people as possible. However, I do think another important point to make is that, for example, during the Zika virus epidemic, there was a development and interest and funding for a Zika virus vaccine. But oftentimes in the setting of emerging viruses, once virus transmission begins to slow, that interest in funding can also dry up. And so many of these vaccines for emerging viruses can often get stuck at different stages of this pipeline before they're actually licensed. Next, I wanted to talk about what these COVID-19 vaccines are targeting. So this is my artist rendition of a COVID-19 viral particle. And I think as many of you have seen all of the really great pictures of COVID-19, you see that there are these sort of spiky proteins that are coming off of the envelope of the virus. And this is actually how coronaviruses got their name. Is that these spike proteins looked like a crown around the virus. And this spike protein is very important because binding of this spike protein to a specific receptor on cells leads to the entry of the virus into the cell and its ability to infect these cells. This receptor is called the ACE2 receptor. It's the same receptor that the original SARS virus also uses. And interestingly, some recent studies have shown that COVID-19 can bind to this ACE2 receptor more strongly than SARS could. And this may be why it's such a good respiratory virus. The first vaccine strategy that I want to talk about today is the development of an mRNA vaccine. And this is being done in collaboration with the National Institutes of Allergy and Infectious Disease and a biotech company, Moderna. The concept behind this is that in our cells, DNA is translated to RNA, which serves as a middleman, and can be translated into proteins that have multiple functions in the cell. Here, they would develop a messenger RNA that specifically encodes the spike protein for COVID-19. This mRNA can then enter into the cell and be translated multiple times by our human cells and the machinery that our human cells normally use to create proteins. Once the spike protein is made in the cell, it can then be shown on the surface of the cell. And because this is a foreign protein, our bodies will then will see this protein and develop antibodies to this protein, which then will be able to neutralize the virus if we're exposed to it. The messenger RNA vaccine is called mRNA 1273 and is currently in phase 1 clinical trials. It's enrolling 45 healthy adults between the ages of 18 and 55 over a 6-week period. And the first patient received this vaccine on March 16th, so we should be hearing some news about this vaccine fairly soon. During this clinical trial phase, they plan to study the dosing, safety, as well as the immune response of the healthy patients. The next vaccine strategy that I'll talk about today is a little bit complicated, so please let me know if you have any questions about how this works. It's called a non-replicating vector vaccine strategy, where an adenoviral vector serves as a mule to transfer information into the cell. And in this case, the information it's transferring is the DNA sequence to the spike protein for COVID-19. This adenovirus can enter into cells and deliver the spike protein DNA sequence into the nucleus, where it can be transcribed into mRNA, and that mRNA can then be translated into the spike protein, which then can come to the surface of the cell, where it interacts with different immune cells, allowing the host to develop a strong immune response to the spike protein and neutralizing antibodies. One vaccine that uses that strategy is called the Ad5-nCoV, and it very recently moved into phase 2 clinical trials, but its initial phase 1 clinical trial planned to enroll 108 healthy adult volunteers between the ages of 18 and 60. And that trial was started on March 15th. And here they're also studying the safety and efficacy of the vaccine to create an appropriate immune response. Another vaccine that uses the non-replicating vector strategy is a vaccine coming out of Oxford University called the ChAdOx1 nCoV-19. And that's currently in phase 1/2 clinical trials. They plan to enroll up to 510 healthy adult volunteers between the ages of 18 and 55. They already started enrolling also in March, and this is a multi-center study. So again, it'll be great to get information about this vaccine in multiple different people from different places. And lastly, I'd like to talk about a concept that Dr. Anthony Fauci touched on in a press briefing, and that's the concept of immune enhancement. I study dengue virus, which is well known for causing this phenomenon. In dengue, there are 4 serotypes or strains. And when you're infected with the first strain, you often will make a good antibody response to that specific strain. However, if you're infected with a second strain, your body sees something similar and begins to make antibodies that can bind to the virus. However, instead of neutralizing that virus, this antibody virus complex can bind to cells, and the antibody then facilitates its entry into the cell, leading to a higher viral load as well as more severe disease. So the idea of immune enhancement will be something that those who are developing COVID-19 vaccines will be focused on during the safety evaluation of their vaccines. So when will we have a usable vaccine? Most experts have mentioned this will likely take at least 12 to 18 months. However, some of those who are part of the vaccine race have mentioned once evaluation of the safety of this vaccine has been performed, it may be feasible to disseminate the vaccine in certain special populations, such as healthcare workers that are exposing themselves to people who likely have COVID-19 on a daily basis. Also, importantly, to make this timeline as efficient as possible, it will be necessary to start upscaling the production of these vaccines as soon as possible. So, what can we do to help move this process forward? If you're living in Seattle, you can go to this website if you're interested in participating in the clinical trial. They are looking for healthy volunteers. But I think also, very crucially, it will be important for us to start thinking about our attention span for these emerging viruses. Preventative strategies can be incredibly useful in slowing and stopping the transmission of viruses. And as these emerging viruses begin to crop up more and more, I think we all as a nation should start thinking about what our investment will be in the future. And with that, my next episode is going to be about diagnostics. I've been getting a lot of really great questions about what are good available tests, why are there so many false negatives or positives, and can you actually get reinfected with the virus? Thank you all for your time and attention. I'm just going to leave a few references here for you if you're interested. Please subscribe and leave any questions below. Thanks.